A very good morning to all of you. So today we will continue our yesterday's topic of discussion, that is biosensor. This is one of the most important topic in instrumentation in paper 1, frequently asked in many university exams. Okay, so yesterday already we have discussed what is biosensor and what are the advantages and applications. Today we will discuss what is the principle, what is the working principle, and what is the instrumentation of this biosensor. Alright, so what is the principle? The principle on which biosensor works is that of your signal transduction. Okay, so signal transduction is the principle on which whole this biosensor is based. And this is the simplified form, your instrumentation of your biosensor. First thing is there. What is first thing? First thing is that of analyte. Analyte is what? Analyte is what? Whose concentration you are willing to measure. So you will write whose concentration is to be measured. Like you want to use, suppose you are using the optical glucose biosensor and you are willing to measure the concentration of glucose. For example, just take the example of your glucose. Okay, or you are willing to measure the concentration of any drug, etc. Analyte can be there, anything is there. So first in instrumentation, first thing is your analyte. Analyte whose concentration you are willing to be measured. Okay. Second thing is your bioelement. This is your bioelement, and this bioelement is also known as your bioreceptor. Okay, so what is the function of this bioelement or bioreceptor? It will interact. What is that? It will interact with analyte. Okay, first there is analyte, and then this analyte will enter into your this bioelement or bioreceptor, and there is an interaction or bounding of this analyte with your bioreceptor. Okay, and this analyte can be your enzymes or any antibiotic, anything. It depends what thing you are going to be measure. Okay, what is the third thing? Third thing is that of your transducer. What is the function of this transducer? This transducer is basically a what? This transducer is basically a energy converter. Energy converter. As when this analyte bound with your bioreceptor or bioelement, then what kind of signal will be there? It gave you a biological response, because these are the biological thing. Yesterday already discovered biosensors terms was biological system, and sensor means senses or detect something. Okay, so when analyte and your bioelement or bioreceptor bound to each other, then after bonding they will generate your biological response. But you know it very well, you cannot measure or you cannot display your any biological response. So that biological response should be converted into your electrical form. That then only it will be visible on your display. Okay, so this transducer act as an energy converter, and it will what? It will convert your biological. It will convert your biological response to your electrical response, or you can say electrical response, or you can say your biological energy is converted into your electrical energy. All right. Next part is that of amplifier. What is the function of this amplifier? As the name suggests, amplifier. That means it will amplify the signal. Simply, what is the function of this? This will amplify the signal. So when first there is an interaction of this analyte and bioelement, and there is a generation of your biological response, then this signal will enter into your transducer, and the function of transducer is it will act as an energy converter. So it will convert your biological response into your electrical response, and after that this electrical response will enter into your amplifier, and the function of amplifier, it will amplify the signal so that you can easily get more signal, and it's visible to display on this display. And what is the processor? The function of this processor, it will receive signal and give processing, and finally display. Okay, and finally display. So processor means processing something, as the name suggests, processor. So what is the function of processor? It will receive the signal from this amplifier, the electrical signal received from this amplifier, and it will processing that signal, and after processing it finally makes such kind of signal that will display on your display, and from this display we can get the what? You will finally get your final reading. Okay, so this is the instrumentation, whole instrumentation of your biosensor. So biosensor asks as a short note. So in short note, what first thing you will write? You will write introduction. What is the definition or what is biosensor? After that, you will write the advantages. Yesterday already we have discussed what are the advantages of your biosensor. After that, you will write types of biosensor. Yesterday already I also discussed that what are the different types of biosensor. So first thing is introduction, second your advantages, then your types of biosensor, then your applications of biosensor. After that you will write the principle, on which principle this biosensor will work, and you will make this diagram and define all these things individually as an instrumentation. Okay, so this is the complete answer of your biosensor, and it is one of the must read topic in paper 1 as an instrumentation. I hope you will like the video. Thank you so much.